All right, so here we have meiosis. So we'll start with a diploid cell, which is an egg or a sperm. Um, and so we go into prophase 1, which is the spindles start to form. So here the spindles over here, and then the homologous pairs, or the chromosomes, will condense in the nuclear envelope here. And then they'll condense, and then the nuclear envelope here will it'll disappear, it'll break down. So now we have the condensed chromosomes and the spindle fibers over here. And so if you can see these black lines here, these are the alleles on the homologous pairs. So these alleles are coding for like hair color or something, and these are coding for eye color. And then also in prophase, we'll have this process called crossing over, where some of the genetic information from each chromosome will share to the others. So like these two will be flipped. Whoa. And so you can see the genetic information has been shared. So there's some of this chromosome, like the orange on this blue, and some of the blue on this orange. And the same thing will happen to these ones. So there's a better spread of genetic information between these chromosomes. And now we move on to metaphase. So in metaphase, the chromosomes will line up in the middle here to be pulled apart, and the spindle fibers will attach. Ooh. Okay. Now attach. So the chromosomes line up on the metaphase plate. And so, alright, so hold up. Yeah, so the independent assortment is like how these chromosomes and their information are distributed into the daughter cells, which this diploid cell will eventually form. And so then we move into anaphase, where the homologous pairs are broken down or segregated, and they're pulled to the poles. So they move down, and the spindle fibers break down, they're pulled to the poles, and they're segregated apart. Okay. So now we have just one of the homologous chromosomes on each side, and one pair on each side. So then we move on to telophase, where the nuclear envelope begins to form back up, and it will enclose the new chromosomes in each of the nuclear envelopes. So the chromosomes then recondense, and the cleavage furrow begins to form. So, these are on the wrong side. But you can see that the cells are starting to be pulled apart, and then we move into cytokinesis, where the cells are pulled apart. So now we have two haploid cells formed, because there's only one side of the genetic information from the initial diploid cell. We now have two haploid cells. So then we go into the second part of meiosis, which is meiosis 2, the prophase 2. Again, the nuclear envelope begins to break down, the chromosomes condense back up, and these spindles form. So, kind of long. So we have spindles attaching to each side and connecting to the poles. It's kind of messy, but you can kind of get the idea. And so then we move on to metaphase 2, or sorry, they're just condensing. So this actually was metaphase, where the spindles attach to the chromosomes, and the chromosomes line up at the metaphase plate in the middle of the cell. So as you can see, there's more genetic information on each side due to the crossing over. And then we'll move into anaphase, where the chromatids are pulled apart, or So each of the chromosomes are pulled apart into sister chromatids, which are pulled apart. So one sister chromatid is on each side, and then the spindle will break down again. So the spindle's gone, and we have one sister chromatid from each chromosome on the side, on each pole. And so it's important to remember that the each sister chromatid is now its own chromosome. Can't pick it up. Okay. So now we've gotten rid of all the spindle fibers, and each one of these chromosomes is broken up into sister chromatids at the poles of each cell. So now we can move into telophase 2, where the chromosomes will recondense on each side, and the nuclear envelopes will form again. Ooh. Yes. So here we have our nuclear envelope encasing that side, and encasing this side. Same thing happens in the other donor cell. Nuclear envelope and nuclear envelope. So now we have almost more daughter cells. So again, the cleavage furrow begins to form, and these cells will be pulled apart again. So So in cytokinesis, the cells will, each of these will break down into 2 more cells. So the total of 4 daughter diploid cells, or haploid cells, my bad. So here's 1, 2. Okay, so the sizing of the cells is a little off, not to scale. Okay, so. Okay, so we have 1, 2, 3, 4 daughter cells. So now there's a total of 4 haploid cells from the initial diploid cells. And so in an egg, this whole process, only one of these offspring would be viable. So like only this one would be viable, and all the rest would be reabsorbed to be reused. And then in a sperm, or spermatogenesis, all 4 would be viable, which is why there's so many more sperm than egg cells. All right, thank you.